Welcome students. Today we are going to study about a genetic disorder, and that genetic disorder is known as the hemophilia. Basically, hemophilia is also called as bleeder's disease. Sometimes it is also called as royal disease, and it is also called as the Christmas disease. First of all, we must know that why it is called as bleeder's disease. It is called as bleeder's disease because here no blood clotting occur, no blood clotting occur, or we can say that blood clotting is prolonged blood clotting occur. Basically, you must know that in normal persons, blood clotting time period is between 3 minutes to 6 minutes. I am talking this about the normal persons. But what about the persons who are actually suffering from the hemophilia? In the hemophilic patient, no blood clotting occur, or if clotting occurs also, then it is prolonged blood clotting. Okay, means by a minor injury, no clotting occur, or if clotting occurs, it is prolonged. Okay, and due to the excessive bleeding, sometime death may also occur. So it is called as bleeder's disease. Why? Because blood clotting is delayed in these persons who are suffering from hemophilia. And why it is called as bleeder's disease? Because the person is actually lacking some blood clotting factors, right? In a normal person, blood clotting factors are present, but there are some important blood clotting factors which are absent in the hemophilic patients, and that's why excessive bleeding occurs. Okay, what happens in the hemophilic patients? Excessive bleeding occurs, and due to this excessive bleeding, sometime death may also occur. But now you must also know about that how many types of hemophilias are there. So hemophilias are basically falling in two categories. One is known as hemophilia A, another is known as hemophilia B. Okay, hemophilia fall in two categories, hemophilia A and hemophilia B. Hemophilia A is caused due to non-formation. It is caused due to non-formation of blood clotting factor, that is 8th. There are many blood clotting factors, but out of that there are few blood clotting factors which are very important for the blood clotting. Okay, so hemophilia A is caused due to non-formation of blood clotting factor 8th. Okay, and this hemophilia A is also called as classic hemophilia. It is also called as classic hemophilia. Okay, and this hemophilia B is also called as Christmas disease. It is also called as Christmas disease. Okay, I have written here that hemophilia is also called as Christmas disease. Okay, so first of all, you must know that hemophilia B is caused due to non-formation. It is caused due to non-formation of blood clotting factor 9th. Okay, because of the non-formation of the blood clotting factor 8th, hemophilia A is caused, which is known as the classic hemophilia. But hemophilia B is actually caused due to non-formation of blood clotting factor 9th. Now the question arises that why hemophilia B is called as the Christmas disease? It is called as Christmas disease because it was first seen in Stephen Christmas. This is the name of the patient. This is the name of the person in which the disease was first of all seen. Hemophilia B was first of all seen in Stephen Christmas named person, and hence the hemophilia B is also called as the Christmas disease. Okay, now this hemophilia was first discovered by John Otto. It was first discovered by John Otto. So hemophilia was discovered by John Otto. So again I am repeating, bleeder's disease, it is hemophilia is called as bleeder's disease because here there are lack of some blood clotting factors, especially 8th and 9th blood clotting factors are absent in these patients, and due to which blood clotting doesn't occurs, or if occur, prolonged blood clotting occurs and excessive bleeding occur, due to which sometimes the death of the person can occur. Hemophilia is of two types, hemophilia A and hemophilia B. It is caused due to lack of blood clotting factor 8th, it is caused due to lack of blood clotting factor 9th, right? It is called as the Christmas disease. Hemophilia B is called as Christmas disease because it was first seen in Stephen Christmas named person, and hence it is called as Christmas disease. Hemophilia was first discovered by John Otto. Okay, so very first was about the bleeder's disease. Now the question arises that why it is called as royal disease. Why it is called as royal disease? So you must know that royal disease, the Queen Victoria of England, Queen Victoria of England was carrier of hemophilia. Okay, Queen Victoria of England was carrier of hemophilia. Okay, and because she was actually belonging to a royal family, and hence it is also called as royal disease. She transferred gene of hemophilia to his son Leopold, and then the disease was spread to royal families of Spain, Germany, etc. Germany, okay, Spain, Germany, Russia, etc. Okay, because this disease was associated with a royal family of England, Spain, Germany and Russia, that's why it is also called as what? The royal disease. So we have talked about these three terms: bleeder's disease, royal disease and Christmas disease. Now coming to this, that hemophilia is a genetic disorder. Okay, so hemophilia is a sex-linked recessive trait. Hemophilia is a sex-linked recessive trait. What does it means? Means gene for hemophilia, gene for hemophilia is recessive. Gene for hemophilia is recessive, and it is found on X chromosome only. It is found on X chromosome only. Okay, it is not associated at all with the Y chromosome. The gene for the hemophilia is recessive and it is found on X chromosome only. It is not at all found on Y chromosome. Okay, so now you must know that how this gene is expressed in the future generations. Okay, so see here I am writing 5 case studies here: XX, XH, X, XH, XH, okay, XY and XHY. Okay, these are the 5 conditions. Okay, small h is here denoting what? The recessive gene, okay, for hemophilia. Now XX, suppose the females are having XX genetic constitution, so such a female who is not having the gene for the hemophilia will be called as normal female, Now a female which is having one X chromosome with a hemophilic gene and another X chromosome is normal, not having the hemophilic gene, so such a female will be called as a carrier female, right? Such a female will be called as carrier female. A carrier female is itself generally normal. Okay, so a carrier female is itself generally normal, but she can have menstrual disorders. She can have menstrual disorders, that is heavy flow of blood can occur during menstruation. Okay, normally she is normal, okay, but she can have menstrual disorders, that is heavy flow of blood can occur during the menstruation, otherwise she is normal. Okay, now XH, she is a hemophilic female. Okay, but this hemophilic female is rare, generally it is rare. Now male is having one X chromosome only, the another chromosome is called as Y, so such a male will be called as a normal male, and the another male who is having one X, okay, but on that X he is having the hemophilia gene, then such a male will be called as what? Hemophilic male. So you can see here that a female can be normal, that she can be carrier, she can be hemophilic, but male can be normal or that can be hemophilic, but a male cannot be carrier. Why it cannot be carrier? Because the male is having only one X chromosome. Okay, so these are the conditions, right, where you can see that the gene hemophilia, that the gene for the hemophilia can cause what? The hemophilia. Okay, if the gene H is present on the X chromosome in male, it will be suffering from the hemophilia, and if the female is having the hemophilic gene on both the chromosome, then the female will be hemophilic, and if the female is having one small h, that is known as the hemophilia gene on its X chromosome, then she will be the carrier of the disease. Okay, now see here that how the disease is spread from one generation to another generation. Okay, I will be talking about three case studies. Number one case study, case number one. Say for if a normal female having the genetic constitution XX marries with a hemophilic male. Okay, hemophilic male is having the genetic constitution XHY, then what will be the condition in the next generation? You can see that XH, okay, X, this is crossed with this XHX, then XY, this with this, then XHX, and then X or Y. I have crossed this one. Okay, so I am encircling the male so you can view the males. Okay, now this is what? A normal male. What about this male? This is also a normal male. What about this? She is a carrier female and she is also a carrier female. Means we can see here that when a normal female is married with a hemophilic male, then the progenies which are produced are, what is the result here? Here what is the result? That when a normal female is crossed with a hemophilic male, then 100% females are carrier. 100% females are carrier. 100% means here you can see that all females are carriers. Okay, and the another result is that all males are normal, they are not at all hemophilic. So this was the result of the first cross. Now talking about the second case study. Say for if we reverse the cross, say for if I say a carrier female, say for a carrier female is a female who is having one that is the hemophilic gene, one hemophilic gene on X chromosome and the another X chromosome is normal. So this is a carrier female. When a carrier female marries a normal male, now normal male is having the genetic constitution XY. So what will happen now? XHX, XHY, we are crossing XX and X and Y. Now I am encircling the males so you can view the males and the females separately. Now see here, this is a hemophilic, this is a hemophilic male and this is a normal male. Okay, she is a normal female not suffering from hemophilia, and as well she is not a carrier. And see here that what is this XHX, so she is a carrier female. After watching this cross you can give the result also. The result is that here 50% males are hemophilic. Means what? Out of total males, why 50%? Out of the total males, half are normal and half are what? Hemophilic. So we can say that 50% males are hemophilic, 50% males are, come on, normal. Means out of the total males, half are normal and half are hemophilic. Now coming to the third one, that what about the females? So we can again, out of the total females, 50% females are, come on, carriers. 50% females are carriers. Okay, and rest is 50% females are normal. So this is the result of this type of the cross. Okay, so first cross I have made between a normal female and a hemophilic male. Now I have changed it, I have reversed the cross, I have taken carrier female and I have crossed with a normal male. So the progenies are of like this type. So this is the result of this cross and this is the result of this cross. Okay, now there must be, there may be a third case study also. Here I am taking just now the carrier female. Say for, if a carrier female, this is case number 3, if a carrier female marries with a hemophilic male, then what will be the case? See, a carrier female is XHX, okay, and hemophilic male is XH and Y. Now cross it, XH, 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 Y, XH, X, X and Y. I am encircling the males so you can view the male separately and the female separately. Now see here, now hemophilic, such a female will be hemophilic female. Why? A hemophilic female is a female who is having the hemophilic gene on both the X chromosome. Now you can see, you can watch here that the female is having the hemophilic gene on both the chromosomes. So she will be hemophilic, and such a female is little bit rare, and it will be rare. And here the female is carrier, here the female is carrier, here the male is hemophilic, here the male is hemophilic, and here the male is normal. So you can see here that for the production of the hemophilic female, the mother must be carrier and the father must be hemophilic, then only the hemophilic female can be born. Now after viewing this cross, you can get the result that out of the males, 50% males are hemophilic. 50% males are hemophilic. Out of the total males, what you are viewing here, that 50% are hemophilic and rest 50% males are what? Are normal. Okay, what about the females? That 50% females are what? Hemophilic. 50% females are hemophilic, and the 50% females in progenies are what? Carriers, are carriers. Okay, so these are the 3 case studies. Based on these 3 case studies, you can solve any problem related to the hemophilia. Okay, now the question arises that what is the reason behind the hemophilia, that why these genetic disorders are caused, so you must remember this thing, that genetic disorder hemophilia is basically caused due to mutation. The potent force which is working for this genetic disease is mutation. Okay, and you must remember this thing, कि though it is called as the royal disease and it was concerned with the royal families of England, Spain, Russia, Germany, etc., but you can also see that this hemophilia is now very common in India and other countries also. This hemophilia is a very common phenomenon, and many person around you you can find that they are hemophilic. So you don't have to believe that always it is concerned with the Queen Victoria family only, or it is actually related, this disease is related to the royal family of the England or the Russia or any other place. Okay, it is a common disease, it is found world over, and it is a genetic disorder, and it is caused due to mutation. Now what to do? The question is what to do to stop bleeding in a hemophilic patient. Okay, so we can give anti-hemophilic globulin injection, anti-hemophilic globulin injections. Anti-hemophilic globulin injections can be given to patient to stop bleeding. Okay, so anti-hemophilic globulin injections can be given to the patient to stop bleeding. Okay, so this was all about the hemophilia. Keep watching. We will be talking about one more X-linked recessive trait known as the color blindness in the next video. Thanks a lot.